have black hoodie stain swabs, black hoodie swabs, pink bodysuit stain swabs, pink bodysuit swabs, as well as Najai Harris blood card.
Okay. Now, what kind of testing did you do in record number 8?
The same as the previous records.
Okay, and that's autosomal DNA?
Yes.
Okay. Now, going through this, you were able to have a sufficient amount of DNA for comparison for Najai Harris's blood card?
Correct.
Okay. And in your first or your second interpretation, this is as it relates to black hoodie stain swabs, but this is the 5AD stain swabs. What were your hypotheses here?
So I determined that there were 2 individuals in that black hoodie stain swab, the first one, 5AD, and I would compare each of the known individuals to the sample. So initially it would be Jarvis Butts, and on one unrelated unknown contributor, and then on the other hypothesis would be 2 unrelated unknown contributors. So based on those results from the black hoodie stain swabs, it was approximately 460 times more likely if it originated from Jarvis Butts and an unknown contributor than if it was from 2 unrelated unknown contributors. And this provides moderate support that Jarvis Butts is a contributor to the black hoodie stain swabs.
Okay. And you can't say what that stain was, can you?
No.
Okay. And then you said you did one for Jarvis Butts, then you did one for Najai Harris.
Correct. So when comparing Najai Harris, it's approximately 3.7 million times more likely if it originated from Najai Harris and 1 unrelated contributor than if it were from 2 unrelated unknown contributors. And this provides very strong support that Najai Harris is a contributor to the black hoodie stain swabs.
And in interpretation 3, this is the second of the black hoodie stain swabs, 5BD. When they're separated like that, when you have like 5BD and 5AD, does that tell you that they're different parts that are being swabbed, different areas?
Generally, it will be 2 different stains on the same item, but the person that actually took those swabs, they would best be able to describe where they were collected from.
Okay. So as it relates to 5BD, the black hoodie stain swabs, what did you find there?
That was 1 individual in that sample, and it genetically typed as female.
And so if that's the case, what was your hypothesis then?
So comparing Najai Harris to that sample, it was 3.6 octillion times more likely if it originated from Najai Harris than if it was from an unrelated unknown contributor. And this provides very strong support that Najai Harris is the contributor to that black hoodie stain swab. And then I compared Jarvis Butts as well, and he's excluded from it to his single source samples.
Okay, and would that be because you found that it was only from 1 individual genetically typed as female?
Correct, yeah. When comparing him, he was excluded.
Okay. And when you say octillion, I'm not a math person, how many zeros is that?
That is 27 zeros.
Okay. And in interpretation number 4, we have just black hoodie swabs, and that's 5CD. And you have two analyses here, one with Jarvis and one with Najai. What did you find as it relates to the comparison with Jarvis?
So the black hoodie swabs, and generally these are taken to see who may have possibly worn it, or skin cells opposed to stain swabs. So I determined that this was from 3 individuals. There were 3 individuals in that sample, and at least one of them genetically typed as male. And when comparing it to Jarvis Butts, it was approximately 2.1 million times more likely if it originated from Jarvis Butts and 2 unrelated unknown contributors than if it originated from 3 unrelated unknown contributors. And this provides very strong support that Jarvis Butts is a contributor to the black hoodie swabs.
And what about the comparison to Neziah?
When comparing Neziah Harris, it was 97 sextillion times more likely if it originated from Neziah Harris and 2 unrelated unknown contributors than if it originated from 3 unrelated contributors. And this also is very strong support that Neziah Harris is a contributor to the black hoodie swabs.
And how many zeros after your sextillion?
Sextillion is 21. And just for reference, anything over 1 million is considered very strong support.
Okay. And your next interpretation is to the pink bodysuit stain swabs. And you performed this assuming that there was 2 individuals. What was your comparison as it relates to Jarvis Butts?
When comparing to Jarvis Butts, it was 6.3 times more likely if it originated from Jarvis Butts and 1 unrelated unknown contributor than if it originated from 2 unrelated unknown contributors. And this provides limited support that Jarvis Butts is a contributor to the pink bodysuit stain swabs.
Okay. And as it relates to Neziah Harris?
It is approximately 60 septillion times more likely if it originated from Neziah Harris and 1 unrelated unknown contributor than if it originated from 2 unrelated unknown contributors. And this provides very strong support that Neziah Harris is a contributor to the pink bodysuit stain.
How many zeros in septillion?
Septillion is 24.
And then you interpreted the pink bodysuit, just regular swabs of it. And you determined that there was 2 individuals, at least 1 was typed as male.
Correct.
Tell me your comparison with Jarvis Butts.
So it was 24 trillion times more likely if it originated from 2 unrelated unknown contributors than if it originated from Jarvis Butts and 1 unrelated unknown contributor. And this provides very strong support that Jarvis Butts is not a contributor to the pink bodysuit swabs.
And you had said previously, just the swabs of items, you said they're more related to like a wearer ID type thing?
Generally, if it's a possible stain, blood stain, semen stain, it'll have the word stain. When it's just swabs of, generally it is just swabs looking for who was possibly wearing that item. But I did not take the swab, so that person would be able to tell exactly.
Okay. And for the comparison with Neziah Harris, what did you find there?
It was 150 sextillion times more likely if it originated from Neziah Harris and 1 unrelated unknown contributor than if it was originated from 2 unrelated unknown contributors, which is very strong support that Neziah Harris is a contributor to the pink bodysuit swabs.
And I think we said previously that was 21 zeros.
Sextillion, yes.
Okay. And what were your findings as it relates to interpretation number 7 of the fabric mattress pad stain cover?
So in the earlier report, we did not have a known sample from Neziah Harris, so I went back and compared her to those previously submitted evidence samples, one of which was the fabric mattress pad cover stain, which she was excluded as a contributor to that stain. Also,
Yeah, you can do.
also to the plastic mattress cover piece swabs, which if you'll remember that was 4 individuals. When comparing her, it was 430 million times more likely if it originated from 4 unrelated unknown contributors than if it originated from Neziah Harris and 3 unrelated unknown contributors. And this was very strong support that Neziah Harris is not a contributor to that plastic mattress cover piece swabs. The final was the shower drain swabs. That was also 4 individuals. And that was 13,000 times more likely if it originated from 4 unrelated unknown contributors than if it originated from Neziah Harris and 3 contributors. And that was strong support that Nozadi Harris is not a contributor to those shower drain swabs.
Okay. And nothing else at the time, Judge.
Okay, Ross.
Good afternoon. So when you receive the swabs, you said in separate tubes, each swab or cutting or whatever you would be testing comes in a separate tube,
Correct.
and it's all labeled with a number that corresponds with the file that you're working on. Is that correct?
Correct. Each one has its own ID, yes.
Okay. And what other communication do you receive with those swabs?
We have submission paperwork, and that is in our system loaded, as well as all of the worksheets from the analysis that we go through. There's chain of custody. I mean, all this is within our system, but we only receive that initial information when we submit it, that paperwork. Occasionally we'll have a police report or something, but in this case I didn't see one.
You didn't have one in this case?
I did not have one in my case file.
Did you have any communication with Sergeant Shannon Jones before you did the testing?
No, I didn't.
Okay. Did you know, was there a name associated with this file that you knew of before you did the testing?
There are, like I said, names within the case file. Generally, we process samples in batches, and we don't really look at all that before we get started until we sit down to actually write the case, and those profiles are already complete.
Okay. And I'm going to try to get some information from you just on how that process works, because I have a limited understanding of DNA, so correct me when I mess up. But as I understand it, when you're running these samples, essentially you come up with, ultimately for interpretation, kind of a line that shows peaks, and that is those are the things that you're comparing. Is that fair to say?
So we look at 24 different areas within the DNA, and 2 of those will just give me the sex of the individual, if they're male or female, if there's a male included. On the other 22, it will be 2 like peaks basically at each location, and you'll have either 1 or 2 each individual.
And there could also be other peaks. So typically you have some that are taller than others, but you could also have shorter peaks. Is that fair to say?
If there's multiple people in that sample, I'll see maybe several peaks, and that's how I would determine how many individuals are in that sample.
Or things that you might interpret as, like, I think they call it stutter.
We possibly, it's a kind of a normal part of the system. There are these peaks that are expected, and they generally are filtered out. Occasionally they get a little high, but we know what they are. So.
And I was going to say, so then when you see this chart, is that part of your interpretation to kind of exclude that as something of significance?
The starter peaks?
Yes.
Yes. I would evaluate if any of those peaks could possibly be stutter or not.
Okay. And I think you answered this, but I just want to make sure. How many loci did you test?
There's 24 total loci.
And that's what you tested?
Correct.
Okay. And that's of like the 3 million base pairs that exist, fair to say? I mean, there's 150,000 loci in the human genome, correct? Is that right?
Yeah, this is the standard for forensics testing, yes.
It's 24. And so I think you indicated in almost all the things that you tested, they were mixed samples, except for the 2 initial, the buccal swab and the ID card. Is that fair to say? More than 1 contributor?
Yes, except for the mattress stain swabs, I believe was a single contributor. Otherwise, yes,
Okay.
more than 1 individual.
On the black hoodie swab, the non-stain swab, I think your report indicates, or your testimony was, that there were 3 contributors. Is that correct?
I just wanted to kind of double check to be
Of
sure.
course. Yep. Are you going to refer to your report to do that?
Yes, my report.
Okay.
So the second, the 5BD,
Yes.
5BD was only one individual, female.
I'm sorry. I guess I'm looking at.
The hoodie swabs, not the stain, you said?
Correct, yes.
Yes, that was determined to be 3 individuals present.
Okay. And I think your report says that at least one was male.
Correct. I can't determine how many males, just that at least one is male because there was a Y in the sample.
Okay. So fair to say there could have been 2 male contributors and 1 non-male, 1 female contributor.
Yeah, all I know is there's at least one in those 3.
Okay. Now, is it the program that determines the statistical number, the probability, or how is that determined?
I determine how many individuals are in the sample, and then the software determines that likelihood ratio.
Okay. Because you're only receiving swabs or cuttings, you don't actually see pictures of what the items are that the stains came from or the swabs came from. Is that fair to say? Or do you?
They are in the case file. I did see them in this case. So sometimes I'll look at them, and sometimes I'll just write the report unless there's a need to see them. They're not always provided, but
Do those pictures indicate to you where the different samples might have been taken from, or the swabs may have been taken from?
yes, the pictures in this case were taken by the previous analyst that provided the samples to me. So she would have actually examined the evidence itself, took the pictures, as well as where any swabs or cuttings were taken.
So the previous analyst would be testing for genetic material in general. Is that fair to say, like saliva, blood, semen, stuff like that? She's the one
Yes.
that's making that determination.
Yes, she would do any body fluid testing, such as blood, semen, saliva, and then any swabs of just for skin cells.
Okay. DNA can degrade over time, fair to say?
Over time and in certain conditions, sure.
Weather, stuff like that,
Potentially as sunlight can degrade DNA.
the elements in general, bacteria.
Yes.
Okay. And so when it degrades, does that mean that the DNA molecule itself is breaking down?
Yes, it's degrading, yeah.
And what is the impact of that on a DNA test?
It could just be lower than it would have been. So there's still a profile, it's just lower. We also see a very specific pattern when something is degraded, where it, instead of peaks kind of being nice and straight, they kind of ski slope off. So we can identify it.
Okay. Could it mean that you get an incomplete or a partial profile?
Sure, yeah.
Was that a factor at all in any of the swabs that you tested in this case?
I didn't see any very specific, that pattern that I was talking about. I didn't see that. Some of the samples are lower, so they may have been possibly, if they were in the elements, degraded or diluted.
Okay. So you mentioned that when you're looking at a profile, if you see a Y chromosome, that indicates to you that there's a male contributor to that sample.
Correct.
And try to say this in the least amount of words possible. And so if you have a mixed sample, you can do, or one of the ways to determine the male profile would be to use a Y-STR analysis. Is that correct?
Yes, we do offer testing that specifically looks at only that Y chromosome in our lab, and I don't perform that testing personally.
Somebody else would perform that?
Yeah, so when we need it, they will perform it, yes.
And what would indicate or what would determine when that would be needed? in your lab?
Generally, it's in samples where it's a mixture usually from a female, let's say a body swab, and there's so much of her DNA, it's overwhelming the small amount of male DNA present. So they can do this testing to only look at that Y chromosome so that that large amount of female DNA kind of isn't in the way.
And so this would be done for something like skin cells or saliva or some DNA, some genetic material that's not specific to males. Is that correct? Like you wouldn't need to do that kind of a test on semen or something that's only going to come from a male.
It's any sample where there's just an a large amount of female DNA, but a very small amount of male DNA is generally when we use the testing.
Okay. Would you use it if there were multiple suspected male contributors?
I don't want to speak to that test, like when we would use it, since I don't do it.
Okay. Is it possible to use that, or would that be helpful in general?
I have no idea.
Okay. You're not familiar with?
No, I mean, I have the basic understanding, but since I don't actually perform it and I'm not trained in it, I don't want to speak to it.
Okay. Is it the Y chromosome DNA, is that passed down to male offspring?
But the male profile, so you're talking about the YSTR profile, it would be the same?
Correct.
Yes.
Okay. From a father to a son?
Correct.
Okay. And that's the Y star DNA, right? The
Yes.
Y short tandem repeat.
Correct, yes.
And so you could have a similar Y star profile between a father and a son because that chromosome is being passed down almost completely unchanged. Is that fair to say? Or is it correct scientifically?
I mean, scientifically, they would have the same, without any mutations, that father and son would have the same Y profile.
Okay.
But in autosomal testing, they would not.
And when you were doing your testing on these samples, you had 2 known profiles, correct?
Correct.
You were testing against Jarvis Butts,
Correct.
yes, and Messiah Harris.
That's correct.
You weren't testing it against any other males.
No other references were submitted to us for comparisons.
Okay. So a DNA test obviously can't tell you when DNA was deposited on an item. Is that correct?
That's correct, yes.
And it can't say how it came to be put there, right?
Correct.
Like it could be there's primary DNA, right? If I cut myself and I get blood on my shirt, that would be a primary transfer, right?
Yes.
If I then touch something and get blood on that, then the blood that's on say it's on my paper, the blood that's on the paper then would be a secondary transfer.
Yeah, like from your shirt?
Yes.
Correct.
Or say if I hand my pen to Mr. Riggins here, and then he hands it to somebody else, that would be a third. Is that correct?
It's the transfer of the DNA. So I touch this, it's primary, then you come and touch it, and my DNA's on it, that would be secondary. And tertiary would be, then that DNA moves again. Of course, each time DNA is transferred, it's lower and lower generally.
Okay. And so then if Mr. Riggins walks away with my pen and leaves it in another room and somebody picks up that pen and uses it, in theory, they could be tested and have my DNA on them, correct?
Possibly.
Even if we were never in the same room together.
Correct.
Okay. Transfer can occur also even like in a washing machine, right?
Possibly.
So if clothes are washed together, which is kind of illogical, right? You're washing something, but in reality, my clothes and Mr. Riggins' clothes could be washed together, and his DNA could transfer onto my clothes just by virtue of being washed in the same cycle.
Correct.
Okay. Wait just a second.
have anything else. Thank you.
Yes.
Redirect?
Just briefly. You asked some questions about YSTR testing. Would it be fair to say that that's kind of like a secondary test you would do if nothing comes up in autosomal DNA testing?
Correct, that's when we look at that type of testing, is when we don't get results with autosomal.
And is that because in autosomal testing would be a direct match as opposed to just a match on a Y chromosome?
Yeah, to a Y profile that
Yeah.
is shared, yeah.
Okay. So in these reports, you had known samples from Nasiah Harris and known samples from Jarvis Butts.
Correct.
Okay. And they were direct autosomal matches to Jarvis Butts.
But we don't use match.
Or-
We
yeah.
would compare them directly, yes.
They were direct comparisons to Jarvis Butts DNA.
Correct.
Okay. And so I believe, as you said, he would share Y DNA with his biologically related male children.
Correct.
But his X DNA would not be the same as his biologically related male children.
No, we get half our DNA from our mom, half our DNA from our father. So that's what makes up your DNA profile.
So if he has a male son, their DNA would not compare, it would only be compared comparatively to the Y chromosomes, not the X chromosome.
Yes, so father and son, they would not have the same profile, but they would have the same Y profile, which is not the testing that was done in this case.
Right. This was direct autosomal comparison.
Correct.
Okay. And is it fair to say that people shed or produce or leave DNA at different rates?
Yes, everyone is different, how much DNA you leave behind when you touch something. Things like blood, semen, saliva, we would expect there to be more DNA present in those types of samples versus just touching something in skin cells.
And given that people can shed DNA at a different rate, if that's kind of the right way I'm wording it, you can't say whether or not something was a primary transfer or a secondary transfer based on the amount of DNA present.
No, I can't say at all how it got there, or even if it's a body fluid. I mean, I don't do any body fluid testing, so I can only say what the DNA is and not where it came from or the type of possible fluids.
Nothing further, Judge.
Thank you.
Thank you. Well done.